right, our next invited author and speaker is Rosie, right? Gradoville. And it's for a paper published in Limnology and Oceanography in 2017 by Gradoville and co-authors. And she is also here giving us an update on the study. Welcome, Rosie.
Thank you. So yeah, my name's Rosie. I'm currently a postdoc in the Zehr lab, and I'm at UC Santa Cruz. And I'm excited to be here today to talk to you about this paper. I'd like to acknowledge my co-authors. And today I'd like to talk about some of our motivations for this research and our major findings, and then spend any remaining time talking about some of the major unanswered questions I think we have in our field and some possible paths forward. So let's start by reviewing why nitrogen fixation is important. Nitrogen fixation is the conversion of nitrogen gas into ammonia. In natural systems, this is just done by the select group of microorganisms called diazotrophs that contain the nitrogenase enzyme. And since this is a really energetically demanding process, we only expect it to be important in environments where nitrogen is limiting. And in marine environments, we think of nitrogen fixation as being dominated by cyanobacteria inhabiting warm, surface, open ocean waters. Cyanobacteria can use energy from the sun to fuel this energetically demanding process, and the new nitrogen that they add to surface oceans can help fuel primary productivity by phytoplankton and support a large fraction of export from the euphotic zone. But some recent evidence has suggested that this traditional view of marine nitrogen fixation could be incomplete. Part of the reason for this is that we might be missing important groups of nitrogen fixers. This is a phylogenetic tree of the NIF-H gene, which encodes a portion of the nitrogenase enzyme. So this shows the diversity of nitrogen fixers. And you don't need to be able to read the names on the tree, but I just want to point out that the green branches are the cyanobacterial groups. So these include the well-studied trichodesmium, but you can see that there are a lot of other groups on this tree. So we know that there is a diverse array of organisms with a genetic capacity for nitrogen fixation. And some recent work doing high-throughput sequencing of the NIF-H gene has shown that, in fact, the non-cyanobacterial groups could be the numerically dominant diazotrophs in many marine systems. We also might be missing some important environments where nitrogen fixation is taking place. This is a map from a study in 2012 showing the global distribution of diazotrophs. But since this study came out, there have been numerous reports of nitrogen fixation happening in unexpected environments. This includes high nutrient coastal systems, and it includes cold polar waters. So it's clear that the biogeography and environmental controls of marine nitrogen fixation are not fully understood. In our 2017 paper, we wanted to address this problem with a cross-ecosystem comparison. So we measured diazotrophic diversity and nitrogen fixation rates in 3 different ecosystems. First, the North Pacific subtropical gyre. This is an area where the cyanobacterial diazotrophs have been well studied. Second, the northwest coast off the United States. This is a coastal upwelling influenced region where we wouldn't expect nitrogen fixation to be important. And finally, the eastern South Pacific on a cruise from Arica, Chile to Easter Island. This included large biogeochemical gradients, and it's an area where it's been difficult to attribute measured nitrogen fixation rates to specific organisms. For each of these regions, we looked at the diversity of nitrogen fixers using high-throughput sequencing of the NIF-H gene, and we measured nitrogen fixation rates using the N15 assay. So first, I want to show you some of our results of the diazotrophic diversity that we saw. This figure shows the relative distribution of different diazotrophic groups across all of our samples. I'm going to walk you through it. Each row in this figure represents a different group of diazotrophs. The top 2 rows are types of cyanobacteria, and all the other rows are different groups of heterotrophs. Each column on this graph is an individual sample, and the larger bubbles represent a larger fraction of total sequences. So right away, we can see really strong biogeographical patterns in our data set. But I want to look at each region individually. In the North Pacific subtropical gyre, we saw that all the samples from 25 meters and 75 meters were dominated by Cyanobacterium eucin A. This is a recently discovered, uncultivated symbiosis between a nitrogen-fixing cyanobacterium and a paramnesiophyte alga. We don't know much about it yet because it's uncultivated, But we do know that the symbiont transfers fixed nitrogen to the host, and the host transfers fixed carbon to the symbiont. We've been able to sequence the symbiont's genome, and it's very small and missing some important genes. So we think this is likely an obligate symbiosis. And you can see that this was the dominant nitrogen fixer across these samples in the North Pacific subtropical gyre. We saw a really different story in the South Pacific. Here we found very few cyanobacterial sequences, and instead it was the heterotrophs dominating the sequences. Something else we found really surprising was that across this wide variety of samples, from coastal Chile to Easter Island, and from the surface down to 400 meters, it was the same group of heterotrophs dominating the diazotrophic community, and we saw this at the OTU level as well. Finally, in the Pacific Northwest, we were able to amplify the NIFH genes, but we saw really high heterogeneity among samples. There were some cyanobacteria, and there were a lot of different groups of heterotrophs present. Now I want to back out, look at all these regions again, and now I'm going to superimpose the nitrogen fixation rates we measured from each region. In the North Pacific subtropical gyre, we found that nitrogen fixation rates decreased with depth. This is what we would expect. And we were not able to detect rates at 200 meters, even though we could detect the NIFH genes there. In the South Pacific, now each bar in this graph is a different sample going from the coast to the gyre. So we found a surprising result with the highest nitrogen fixation rates in the coastal samples where fixed nitrogen concentrations were also the highest. And finally, in the Pacific Northwest, we were not able to detect nitrogen fixation from any of our samples. We actually thought this was one of our most interesting results because zeros are not reported very often in nitrogen fixation research. And we did a sensitivity analysis and calculated detection limits. And the detection limits that we calculated were higher than a lot of reported nitrogen fixation rates from the literature. So we think that this is just a good example of why it's important to report accurate detection limits with nitrogen fixation rates. And I also want to point out that, again, even though we couldn't measure nitrogen fixation rates in the Pacific Northwest, those genes were still there. So one of the overall conclusions of the paper was that NIFH genes and diazotrophs may be nearly ubiquitous in the oceans, but nitrogen fixation rates are regionally restricted. And just because the genes are there doesn't mean this process is happening with a biogeochemical significance. So where does this leave us? To me, there are still some large, fundamental, unanswered questions in our field. What organisms are driving nitrogen fixation rates? What are their environmental controls? And can we use this information to constrain and predict global nitrogen fixation rates? In my postdoc, I've been able to do some work looking at these first two questions, so I want to give you some examples. So first, which organisms are driving the rates? I've been able to go on a couple cruises to the North Pacific since this 2017 paper. Here are a couple maps showing cruise tracks going from Hawaii north through the subtropical gyre and into the North Pacific transition zone. And on both of these cruises, we found strong evidence that UCNA was a major player in nitrogen fixation. One line of evidence is the sequence data. Here you can see pie charts showing the relative distribution or the relative abundance of different diazotrophic groups with UCNA in blue. So in DNA from both of the cruises and also in RNA from the 2017 cruise, it was UCNA dominating the sequences. But this is kind of indirect evidence. It doesn't prove that UCNA is fixing nitrogen at high rates. So I think a little stronger evidence we saw when we compared the abundance of UCNA to community nitrogen fixation rates. And we found really strong relationships. This top graph is from 2016, the bottom graph is from 2017. We have UCNA abundances on the x-axis, nitrogen fixation on the y, and you can see we found very strong relationships. So we think that UCNA is an important organism driving nitrogen fixation in the North Pacific, at least in the springtime. But as I said, UCNA is uncultivated, so we know very little about their environmental controls. We can't run lab experiments to learn how they react to nutrients or light. So I think one really cool tool that we can use, which Mara gave a great introduction to, is nanosims. We can do our standard N15 assays, but then we can use cardfish to stain the UCNA symbiont and host, and we can find those UCNA symbioses and run them on the nanosims to get the elemental composition of individual cells and calculate cell-specific nitrogen fixation rates. in the last year, I've been able to start using the nanosims. And so I want to show you a preview of some of my data. Here I did experiments with light. And each dot on these graphs represents the USNA cell-specific nitrogen fixation rate from an individual symbiosis. On the left panel, I did 12-hour light-dark incubations. And we can see very clearly that USNA only fixes nitrogen during the light period. On the right graph, I did nitrogen fixation rate assays on an in situ array to look at how rates varied with depth. And you can see that USNA nitrogen fixation does decrease with depth, but that we could detect rates still at 100 meters. So I think this is a really powerful tool that can teach us about the environmental controls of USNA, but also about the environmental controls of the heterotrophic groups, which we also have very few cultured representatives of. So ultimately what we want to do is constrain and predict global nitrogen fixation rates. And I just want to leave you with a figure from a paper that came out just this month in Nature, which used 2 different models to predict global nitrogen fixation rates. And you can see that these 2 models agreed very well overall. But one area where they disagreed was the Arctic Ocean. And here the global inversion model they used, which uses geochemical methods, predicted there to be some nitrogen fixation in the Arctic. In fact, Katie Harding, a PhD student from our lab, just published this paper a few months ago showing where she used nanosims again and showed that USNA is in the Arctic fixing nitrogen. So we know that in at least some areas of the Arctic, like the Bering Sea, some nitrogen fixation does take place. But in the study, by weighing it all, the prognostic ocean model doesn't predict any nitrogen fixation in the Arctic. And this is a global ecosystem model which has a group of diazotrophs parameterized based off of cultured representatives like trichodesmium, which have narrow thermal tolerances. So I just think this is a great example of why we really do need to know which organisms are driving nitrogen fixation rates and learn about their environmental controls in order to be able to accurately constrain and predict global nitrogen fixation. So with that, I'd like to thank my co-authors and funding sources, and I think I have plenty of time for questions.